In this video, we will be exploring some of the potential ways in which asthma can be switched off through gene therapy. You may be asking, what is asthma? What is gene therapy? Simply put, asthma is a long-term medical condition that affects the airways, characterized by inflammation, increased production of mucus, and tightening of the muscles around the small airways. This can be triggered by airborne allergens, respiratory infections, and physical activity. And while at its least, asthma can be a minor issue. However, at its worst, asthma can be a major problem that interferes with daily activities and may lead to a life-threatening asthma attack. As such, modern biotechnologies have focused on the prevention of asthma through gene therapy. It's been evident that scientists are trying to provide a medical approach by correcting the underlying genetic problem. Relative to asthma therapy, this technique is projected by the switching off of asthma-causing immune responses via the concept of replacing defective genes with healthy ones. While we do have methods to reduce the impacts of asthma, for example, relievers in corticosteroid-based preventers and oral medication, these work well for the majority of patients. But those with severe asthma, gene therapy could be the key for relief. As such, research into asthma therapy is needed to provide an adequate and long-lasting method of treatment. This is especially relevant for those in lower and middle-class society, where the cost of asthma is approximately $11,740 per person per year in Australia. Similarly, this research is important as people with asthma are more likely to report a poor quality of life. Application of gene therapy has the capacity to reduce the burden on individuals and society by reducing medical costs, hospital visits, and increasing quality of life. While inhalers and medication are effective, those with asthma have limited ways to prevent the next attack from occurring. The three methods of gene therapy discussed in this video would cover the application of thymulin gene, IL-4RA, and dendritic cells. Thymulin gene is used to reduce inflammation as it produces the hormone essential for T-cell differentiation and function. It is involved in anti-inflammatory mediation, and if we target this gene that produces thymulin, we can interfere with its expression, thus inhibiting the flaring of immune response. So here's how it works. In research, it is found that a single dose of nanoparticle carrying thymulin-expressing plasmids will normalize inflammation in asthmatic lungs and improve airway structure and function. However, this research is only currently being carried out in rodents. Hence, there are possible applications for human use in the future regarding the use of the thymulin gene to reduce inflammation. Another area of interest of gene therapy for treating asthma is IL-4RA therapy. IL-4RA therapy focuses on the production of cytokines secreted by allergen-specific type 2 T helper cells. Dysregulated Th2 type cells are understood to cause the clinical manifestation of asthma as an allergy, while cytokines IL-13 and IL-4 are integral for asthma pathogenesis. By targeting these cytokines, researchers discovered that by inhibiting them using a mutant IL-4 protein called IL-4RA will eliminate asthma symptoms from presenting individuals. This process consists of a few steps. Firstly, a recombinant adeno-associated virus AAV vector is created. This is because recombinant adeno-associated viruses are the best for long-term gene expression and positive results when used for the genetic manipulation of diseases. Then the vector is used to deliver and provide sustained expression of IL-4RA directly into the vein of mouse models. The adeno-associated virus delivered IL-4RA is expressed systematically into the airways of the mice after they showed allergy sensitization. This method was tested on murine models where IL-4RA inhibited the development of airway eosinophilia and mucus production in the mice and also reduced the levels of asthma associated with Th2 cytokines. Similarly to the previous technique, at this point in time, this form of gene therapy has only been tested on mice and other murine models. Applications for human use needs to be investigated, especially as within this research, the IL-4RA is administered directly into the airway, which is an invasive procedure. A final gene therapy technique to disable asthmatic symptoms is the genetic editing of bone marrow in order to produce modified immune cells. Here, the change of this immune cell changes the response of others. As such, this alteration results in typical asthmatic symptoms such as coughing, wheezing, swelling of the throat, chest pains, and lung inflammation to be prevented. Bone marrow cells, also known as stem cells, are cells with the potential to develop into many different types of cells in the body. Through this technique, the stem cells from the bone marrow are genetically edited to develop to specific immune cells known as dendritic cells, which are antigen-presenting cells. In current studies, this process of gene editing has been utilized to reduce asthmatic symptoms in asthma-induced mice. So these genetically modified stem cells within the bone marrow are then injected into the mouse, where it becomes a part of the mouse's bone marrow, and will start to produce numerous amounts of the specific dendritic cells. In typical asthmatic responses, the allergens are picked up by B cells or macrophages and presented to T helper cells, which initiate an immune response. This response often works in combination with inflammatory responses caused by memory T cells in the body. As dendritic cells present specific antigens on their surface, their alteration to display specific allergens can work to block the adapted T cell response when they interact with helper T cells. This leads to a treatment of the asthmatic conditions as due to the allergens no longer being detected as a threat. The typical immune response doesn't occur, and specific memory T cells that cause the asthmatic response become inactive by deletion and no further memory T cell creation. As a result of this technique, airway inflammation is alleviated and airway hyperreactivity is reversed. However, once again, this research has only occurred for rodents, but hints at the development of effective immunotherapies in humans. In conclusion, there are many forms of researched and tested gene therapies that target asthma. Thymulin, IL-4RA, and dendritic cell therapies are all forms of gene therapy that successfully help cure and manage the most common and harmful symptoms that asthma presents. These therapies are able to replace faulty genes or add in new genes in an attempt to help improve the body's ability to fight the disease. However, all of these therapies have only been tested on murine models. Further, as some of these methods are very invasive, extensive research for safe human use is crucial. With asthma being such a prevalent problem for a large part of the population, the advancements of these gene therapies will not only prevent harmful attacks from occurring, but also improve lung function and overall quality of life for many individuals.